All right, thank you for attending today's lecture. I'm Chris Larson. I'm not Dave Arnold, who usually does the introductions. I'm the Associate Vice Chancellor for Research for the campus, as well as the Associate Dean for Research and Graduate Studies in the College of Veterinary Medicine. It's my pleasure to introduce our speaker today, Dr. Yves Chabu, and his title will be Developing a Microbial-Based Immunotherapy Against Pancreatic Cancer. Just as a little bit of background, the Discovery series of lectures is the Next Gen Precision Health Initiative flagship speaker series, welcoming attendees from across academic disciplines and from our community throughout the state of Missouri. Dr. Chabu's work, there are 500,000 new cases of pancreatic cancer diagnosed worldwide each year, and the majority are discovered at an advanced stage. With current therapy options, only 3 to 8% of those advanced cancer patients will live longer than 5 years. Dr. Chabu's work is part of an exciting new subfield involving genetically engineered bacteria that can travel where immunotherapies can't, while also stimulating immune cells. It's an off-beaten-path solution with the potential to develop into important new treatments for patients who have historically had very few options. Dr. Chabu is an assistant professor of biological sciences at MU's College of Arts and Sciences. In 2002, holy cow, how about 2022? The college named him Assistant Professor of the Year. He received his Ph.D. from the University of Oregon and the Howard Hughes Medical Institute, and there he studied the molecular mechanisms that govern aspects of neural stem cells. He subsequently joined the Yale School of Medicine in 2008 for his postdoctoral work and fellowship. This involved the studying fruit fly and mouse models to better understand how mutations in the RAS gene family affect cell signaling and lead to various cancers. He started his lab at MU in 2017 using translational approaches to help explain why certain cancers are resistant to existing treatments. And again, it's my pleasure to welcome him to talk about his work today.
Thank you. Thank you. Thank you so much for that nice and thorough introduction, I should say. And it is really a pleasure for me to be here. I am delighted to see so many familiar and unfamiliar faces, and I hope that this is going to open up sort of opportunities for collaborations with colleagues and so forth here on campus. All right. So I'm not going to go through the title of my talk. It's self-evident. Just sort of by way of introduction, like Dr. Larson said, we have been using different animal model or cancer model to really understand the molecular logic that underlies robustness of tissue growth, right? And that is in a setting of either development or tumor growth, or in a setting of response to therapies. And we've been relatively successful applying this method. So we are integrating sort of these multi-level omics across model systems to gain actionable insight into really the molecular underpinning of tumor growth. So here I'm sort of just have selected a couple papers that really highlight the type of approach that we're utilizing to reveal what we hope will be insightful data that could be used in a translational setting. All right, so the era of precision medicine, we are at Next Gen building. So the era of precision medicine is certainly here. And I should say that with the advances in molecular biology and the following drop in the cost of doing all these all-omic, whole genome sequencing has really catalyzed sort of advances in molecular typing of all these different tumors, right? So today, you can sequence your own genome for just the cost of your dinner, right? So it's just how And I'm not saying you should go to a fast food place that the price might not match, but I think you get the idea, right? So the prices drop dramatically. So the sort of the paradigm that is used to essentially actualize precision medicine goes as follows. So we know that we have basically molecular information on all these tumors, right? Because these tumors have been sequenced. And from those sequences, we can correlate these molecular landscape or these mutational landscapes with tumor progression or tumor response to specific therapies. And so then when patients present with a particular cancer, then there's the molecular profiling that ensues from the biopsies. And based on that information, then we can then stratify patient to a specific therapy. That is, if they have a mutation that is actionable, a mutation against which we have therapeutics. And stratification, we can then eliminate those tumors and ultimately achieve durable survival for these patients. But we know that is a little more complex than that. It's a little more complicated than that. And here I'm highlighting my favorite signaling pathway, the EGFR-RAS. This oncogenic activation in this pathway is associated with really aggressive disease and disease that's resistance to current therapies. So here is a patient that presents to the clinic with these nodules, these melanomas. And again, following the paradigm that I'm showing you to the left, we then collect biopsy from that patient. And for this particular patient, it was established that they had a mutation in BRAF, which is this here. So they had a point mutation that leads to constitutive activation of the signaling pathway. Following that logic, then a BRAF-specific agent was then used to manage this patient, and you can see remarkable response following treatment, right? And this is 4 months following treatment. And I wish I could tell you that's the end of the story. Unfortunately, it is not, because 2 months after that, the patient essentially relapsed, and you can actually see that those nodules come back almost precisely where they were at presentation. So these really unfortunate outcomes, that is just the disease recurrence and resistance, is really limiting the depth, the magnitude, and the durability of response. So by depth, I simply mean we're not, if you have a particular lesion, you're not eliminating all those cells. And also the duration, you just go for a certain period of time and then the tumors will actually relapse. So there are several models that have been proposed to explain where these recurrence or this resistance is coming from. This includes histological transformation, stem cell, right? The cancer stem cell theory. I am not going to go into that space. Instead, I am just going to go into the topic of today's, which is pancreatic cancer. So what I've described to you is really not just akin to, is not specific to melanomas, right? So the same type of challenges occur when you treat patients that have lung cancer, patients that have different other malignancies, using targeted therapy approach. So resistance is definitely a challenge to achieving durable outcome in patients. And I think what illustrates that even better is pancreatic cancer. Like Dr. Lawson mentioned, so the pancreatic cancer is an area of huge clinical unmet need. You have several patients present to the hospital when the disease is already advanced. And for those patients, curative surgery is not an option anymore. So then they simply receive chemotherapy, but not really surgery. So the patient undergoes surgery and chemotherapy, 34% will be alive 5 years from the time of presentation or the time of the intervention, whereas the patient where the disease had already advanced, only 3 to 8% will be alive 5 years post that intervention. I should also note that even patients that receive this surgery with curative intent, they can have micrometastases that actually flare up post-surgery somewhere else. Again, really highlighting a critical problem in this indication. Furthermore, the targeted approach that I showed you earlier in melanoma, that approach is also not available in pancreatic cancer. Here I am showing you, we know that KRAS is the main driver in oncogenic activation of KRAS is the main driver in pancreatic cancer. And a couple years ago, we were really excited to hear that there was a small agent, a small molecule sotorasib, and now adagrasib, which target RAS. So this agent target RAS in a off position. It will bind to RAS in a GDP state, and this will preclude it to go into the activated state. So this has been used extensively, especially in lung cancer setting. Unfortunately, this agent cannot really generate significant or meaningful impact in pancreatic cancer space, and that is because this agent is specific to the RAS V12C allele mutation, right? So, and that specific allele is only present in a small subset of pancreatic cancer patients, accounting for roughly only 2%, okay? And furthermore, from data that we're seeing in lung cancer where this drug is being used, acquired resistance remains a challenge. So patients that are treated with sotorasib or adagrasib ultimately relapse. And here I've just highlighted a couple papers that really bring this point home. So we need essentially better approaches to achieve broad and durable outcome for patients in pancreatic cancer, okay? And we know that immunotherapy is certainly one way we can get there, right? And it will become a little more apparent here in the next couple slides. But before I go into the immunotherapy space, I want to provide some general background. I realize this is a sort of public lecture, so there are some background that will be needed. So if you're an expert in immunotherapy, please hang tight and be patient. We'll get there. All right, so we, like I said earlier, so we know that immunotherapy can achieve, has really transformed the treatment landscape for many tumors. And here I'm just sort of give you sort of a primer on the interaction between cancer cells and immune cells. So here I'm depicting a cancer cells, and as these cancer cells are growing, they present antigens that are then detected by immune cells. And here I'm showing, for example, a M1 macrophage, which will release molecules to try to kill the cancer cell. And on the other hand, this molecule produced here by this macrophage can also stimulate this other type of immune cells called T cells, and they also called CD8 positive T cells. These cells have a cancer killing capability. So what essentially happens, the cancer cells can then produce molecules that will corrupt these cancer-killing macrophages and force them into a tumor-supporting phenotype. So they release molecules, and this become the transition into what's called an M2 phenotype, but that's not really critical for the talk. But this M2 phenotype or these cells can ultimately suppress these CD8 cells, and the cancer then can continue to grow unrestricted. Okay. Now, Another way that the cancer cells achieves immune escape is by producing this molecule at the surface, PD-L1, which interacts with a receptor PD-1 in the T cells and basically turning off that T cell. Again, another mechanism by which cancer cells can really turn off the immune environment so they can continue to grow. Recent advances in immunotherapy have identified sort of strategies where we can go after these interactions, so we can essentially inhibit this T cell exhaustion signaling. And in doing so, we can achieve basically immune-driven cancer cell elimination. And I think this is really illustrated here in the case of melanoma. So we were talking about melanoma earlier. We talked about small agents, right? And we saw that the response was really transient. And unfortunately, the patient relapse and perish soon after that relapse. So here we then, if we use immunotherapy, we can see that we can achieve, so these dots here, these are all this disease. So we know that we can achieve significant really benefits and durable benefits in melanoma, right? So here we can see disease clearance, and this can go on for a couple years. You have patients that actually have done really well under using this modality, immunotherapy. So unfortunately, this type of approach is not available in pancreatic cancer, right? And the reason being is that pancreatic cancers are inherently cold. So the immune environment is suppressive. So these immune cells in the tumor microenvironment, they can't get to the tumor, or when they get to the tumor, there are signals in the tumor microenvironment that suppresses these immune cells, and therefore immunotherapy cannot work in that space. So from talking to colleagues and including clinicians, it seems like the most direct path to achieving benefits or generating durable benefit in pancreatic cancer space will be to convert the pancreatic cancer immune environment from cold to hot. So that is to activate the immune component in the tumor microenvironment, and in so doing, then potentiate all these immune cells which can then go after the tumor. This has benefits not because you can generate localized response to that primary tumor, but also you can generate systemic response in the sense that these immune cells are activated, can then continue to patrol the host to identify and kill residual disease in that patient. So clearly, we need to go the immunotherapy route. So how do we get there? So here, I would like to then really take you on a different path and really non-traditional. And just like Dr. Lawson said, this is really off the beaten path, right? It's really thinking about it differently, right? So in light of all the challenges that we encountered using traditional medicine, by traditional I mean conventional medicine, we need to think about this problem a little differently. We need to be creative, we need to be bold to confront these challenges. So to get into immunotherapy space, I want to bring up the case of William Cooley. So William Cooley was a surgeon, and so he did his training at Yale, then went to Harvard, and from Harvard he went to New York University to practice, which is now Memorial Sloan Kettering. And when he was there and seeing patients, he noticed that patients that were infected with streptococcus, their tumors would actually just dissolve. The tumor would disappear if they had that streptococcus infection. And prior to him, Bush in Germany had a similar observation where if a patient had some infection and they had some inoperable disease, that tumor was able to regress. And in this case, then what he did based on that observation, he actually hunted down a patient of his that had head and neck cancer, so which is this individual here. and found that patient 7 years after they left the hospital, and because they had an infection, right? So they had a streptococcus infection. So 7 years, they were completely okay afterwards, right? So that led him then to really dig through the medical records, and he was able to identify 47 similar cases where patients had an infection unrelated to the cancer and yet had a really nice outcome in terms of tumor regression, and they were able to continue their lives, right? And so this is, here I'm showing just, it's not unique to sort of head and neck cancers, but you can also see here these breast sarcomas. And this motivated Cooley then to initiate sort of some trials and actually start treating patients. He was treating patients now with the bacteria. He was intentionally infecting patients with streptococcus at the beginning to generate what he thought would be some immune activation to then get rid of the tumor. So at the bottom here, I'm showing you again, so these are patients in other head and neck tumors. You can see that these patient actually went on, he was completely cured, and patient went on and lived a happy life. The patient who in the middle here also was responding well to the treatment. I should now say that one challenge with Cooley's therapy was that he really didn't understand the mechanism of action. He knew that if you treat with this bacteria, you're going to get a response in terms of the tumor. But how that was actually happening was really not known to him. Furthermore, there was quite a bit of variability in terms of how patients reacted to the vaccine. So for instance, the gentleman here in the middle, which is the same gentleman shown in the far left, required 63 inoculations before a response. 63 inoculations before a response. Where some other patients earlier, right, lower 10, 5, and you had a really good response. So those sort of that lack of consistency really began to spell trouble for Cooley. And it didn't quite help because at the same time, the chair of his department, Ewing, was really interested in radiotherapy. So there was the rise of radiotherapy, and also some chemotherapy were coming onto the market. And there we understood clearly what radiotherapy was actually doing. We knew what irradiation was actually doing to the tumor cells, right? Chemotherapy similarly. We had some understanding of what was going on. So then the chair at the time, his chair instructed him to cease with his immunotherapy or his bacteria treatment. And so he stopped treating patients with his cocktail and left New York University and died shortly after that in the 1930s. Interestingly, his daughter sort of took over the reign from there, Helen, and she was basically following all the patients that her father had treated. And her along with another surgeon, so they were able to establish that the response rate and magnitude of response was actually comparable to conventional therapy. So we are still in the 1940s, 50s. So I think you get the idea where we so it was a bold idea that he had at the time, right? But we're just not ready for it. Okay, so so this basically just provides you sort of from that point on, from those initial attempts of bacteria therapy against cancer to where we are today. A lot of things have happened, right? So again, like I noted here, Cooley did not know the mechanisms. The outcomes were really variable, and again, radiotherapy came into play. And I'll fast forward here. So if you don't see your favorite or your rock star scientist on the timeline, I apologize. I can't include everybody on there, but these, I think, for me, are really sort of seminal findings that motivated or catalyzed sort of the evolution of immunotherapy. Okay, so fast forward from that point onward, so we know then Tasuku Honjo identified PD-1, which is the molecule I told you earlier. And then Lipin Chin identified PD-1, right? So the receptor, the ligand at on the immune cells. And then right in the early 2000s, Mario Schnolz at Yale with Vion Therapeutics decided to again go back to the bacteria. They wanted to use the bacteria as a vehicle to deliver therapeutic loads in the tumor. So we known at that time, we knew that bacteria have this ability to home to tumors. So if you can leverage the tumor tropism, that is their capability to target tumors and therefore deliver chemotherapeutics, so any agent that would then kill the cancers is definitely a better approach, right? So they started some studies using this strain that's called VNP. I'm just gonna call it VNP, and he went into clinical trials and failed in clinical trials, and primarily because it was not sufficiently attenuated. So from conversation that I've had with Mario, as soon as the patient received the bacteria, it was rapidly cleared by immune component, by immune cells. So they were left with really low titer in the tumors. So they even tried some intratumoral injection in patients, and they had slightly better response, but then they were not stable. It was not really staying in the tumor and stably colonizing that tumor. So, and then fast forward, now we also know that Alison discovered CTL4. I'm gonna skip that part. I hope he forgives me. And then later on in in 2010, so we know ipilimumab and other PD-1 and PD-L1 targeted agents were approved by the FDA. And in 2017, we moved to CAR T cells, so things have gotten a little more complex. And what I want to point your attention to is roughly between 2017 and 2019, the National Cancer Institute expressed interest and strong interest, and again, in going back to bacteria to achieve cancer elimination. So using microbial-based technologies to target cancers. And this is particularly interesting in a setting of metastatic disease, right? So you want to generate that immune activity that can be systemic and then achieve durable benefit. And more recently, the FDA granted fast track designation to Sadkiva, which is basically salmonella that's delivering interleukin-2 in pancreatic cancer. This is after 2 clinical trials. There's a phase 1 and a phase 2, beautiful result. That study read out. You can actually see a really nice response in these patients, so leading to this fast track designation. In other words, it's the Renaissance and we're back, okay? We are back, and what I hope that we can do now is to learn from everything that has happened in the past, including the VNP clinical trials, and what these we're going to learn from Sadkiva now in the clinical development. Okay, so using that information, we know now we can use bacteria. So here I'm showing you the compromise that must be met, and that is in terms of attenuating the bacteria and efficacy. When you talk about salmonella, you say, I'm using a salmonella to treat cancer. The first response that I get often is, are you crazy? Right? So because salmonella, you don't associate salmonella with something nice, right? So we have to really balance the toxicity concern with the efficacy. There's some really nice modeling that were done here by Fegner And you can see that there is a relationship that we need to strike, and that is, we know that if the strain is super attenuated and therefore safe, the efficacy is not that great. So we need then to use current tool, genomic editing, so we can transform a strain that is attenuated but not really efficacious to actually make it really efficacious, right? And so we can do sort of this genomic editing at will, and to then ensure that this strain that we generate, the therapeutic strain we generate, has a behavior of interest, right? So in our case, we want to have moderate immunogenicity. We don't want it to activate the immune system overly, and we want it to be safe. And we also want to make sure that it is tumor targeted, right? And that it is stable. And this is learning again from the VNP clinical trials. So this now brings me square really to our technology. So where does this technology come from? So this was derived first, it was isolated from a collection of mutants that were basically grown under nutrient deprived conditions for 50 years. In so doing, you can see that there is a lot of reprogramming in this bacteria. And so this is natural, it's a natural evolution that actually happened. So that collection of strains was then used to screen, to identify a strain that can selectively or preferentially colonize tumors, leaving untransfected cells alone. So this is an in vitro assays. And based on that approach, this strain 1634 was identified. And again, that had to be de-risked. So there was a RFH deletion, which is an indirect way of reducing the abundance of LPS, right, which is established virulence. So this is a positive regulator of LPS biosynthesis. So by deleting this, we were just not eliminating LPS altogether, but we're just lowering LPS in this strain. Second, additional sort of tumor tropism enabling strategy were then taken. Here we then introduced a mutation in ROA and Thi-A. So RA is all aromatic amino acid, and thymine also was depleted. So this selection was particularly interesting because we know that tumor cells upregulate these aromatic amino acid, and in some cases even thymine. So we thought we can establish metabolic complementation by having the strain go to the sites where these molecules are highly upregulated, so they can preferentially colonize with tumors. And second, we noted that the bacteria that we have has a particular affinity for mannose-linked proteoglycan. And so we showed that this bacteria can bind to cancer cells that have mannose-enriched proteoglycan with 10 to 400-fold more affinity than those that do not have those mannose. So that gives you some targeting between non-specific versus specific. This generated strain 2631. This strain was then evaluated for safety, first in mice and then in healthy dogs, where it proved to be safe, well tolerated from blood chemistry. So you had a typical response following treatment, right? So not feeling good, there's some vomiting happening, but then that quickly subsided, and the dogs were just fine. And then the blood chemistry also showed, from blood chemistry, we could actually see that there was no major organ disease. And then lastly, we then demonstrated that you could actually colonize and stably colonize tumors. So in a paper that we published in 2020, we demonstrate that it goes into the tumors, and once it enters the tumor, it can replicate within the tumor. Okay, And I should also note that all these changes that we did not really affect the ability of the strain to grow in anaerobic or aerobic conditions. This is important because you want a strain that can also go into the core of the tumor where it's highly hypoxic. Right? So we had to make sure that all these modifications, they're not impeding with the desired characteristics. Finally, we then evaluated the genetic stability of our technology in vivo, and that is, we wonder whether once it infected the tumors, if it was genetically stable such that it will not acquire additional polymorphism or additional mutation that will then make it lose these desired characteristics, which are genetically encoded anyway, right? So we did the modeling, and we found that it would actually take 10,000, if I calculate it right, it would take 100,000 days, so about more than 200 years for the strain to develop in a SNP, a mutation, following once it gets into the tumor. Right? So this was done through longitudinal into animals. We collected all these bacteria from the tumors and then comparing polymorphism or genetic evolution across. And we see it would then require, and then we apply some modeling on that. It would take about 200 years after treatment to have additional SNPs. So we can all agree that 200 years would be okay with that. Right? We can live with that. All right, so this strain then was tested against pancreatic cancer cells. So here, this is just an in vitro assay, and we could again demonstrate that the strain can preferentially kill pancreatic cancer cells when compared to control cells, right? So it's really remarkable. Then the next thing then we ask, so this is all in vitro, so can we achieve now tumor tropism? Can we test the tumor tropism and safety of our agent in vivo? So we turn to a pancreatic cancer mouse model. This is a syngeneic orthotopic model where the pancreatic cancer cells are introduced into the pancreas, and that will basically initiate these tumors in the pancreas, and they will grow rapidly. They'll actually generate metastases. They'll metastasize to all the visceral organs, most of the visceral organs, and the animal will die roughly within 2 weeks following treatment, right? So before we could do that, we first thought that we should introduce a marker that will enable visualization of the animal in vivo. So we then introduced a far red RFP here, an infrared RFP into the strain, but we also then attached to that an antibiotic selection, chloramphenicol resistance, an antibody resistance cassette, which was then introduced in the genome, right? Not from a vector, in the genome. So the RFP then allowed us to visualize the strain in vivo, and these cammodulin-resistant cassette allowed us to identify and isolate basically the bacteria from the tumor when they're harvested, and then to derive bacteria count in tissues, right? So using these two sort of orthogonal approach, we can then demonstrate tumor tropism. What I hope you can appreciate, so these animals here to your left at B6, and this are just treated with PBS, so this gives you sort of the background in the assay, some autofluorescence. However, if we treated the animals with our agent here, you can see that the animals that have the tumors really light up, right? So you can actually see the strain in vivo, and whereas the animal that does not is definitely less, right? And again, I want to draw your attention that there is background. We then harvested these tissues and then just look by immunohistochemistry by staining and ask whether we can actually see in the pancreas. So this is straight in the pancreas. And you can see that in the pancreas that basically come from animals that had pancreatic cancer, we can see our agent in red. However, the tissues that did not have cancers, you do not see that RFP. So that's really encouraging. We then derive a quantitative measures of this phenomenon here in F. So we basically counted the number of colony, right, that you see in tissues. Again, you can see that in pancreatic cancers, In pancreatic tumors, it's really high, and then also in metastases, we can also see it. And this is also compared to the liver, which is a clearing organ, so it's not surprising, right? So it will be cleared systemically and will go to the liver. And we know that these treatments are safe because following treatment, here we are treating every 3 days for full treatment. So this is the period of treatment, which normally will go to 9 days. And we don't see any treatment-related lethality compared to our control. So this was really encouraging. Now we then ask whether our agent, since our agent is generating some direct cell killing, as I showed you earlier, and we thought that that could trigger some immunogenic cell death, right? So you can actually activate the immune system based on that killing that you're achieving in the tumor microenvironment. Furthermore, we previously showed that our agent can release all these cytokines and chemokines in the tumor microenvironment, which we thought would then augment the recruitment and potentially the activation of all immune cells against the tumors. So to first assess that in this particular model, we looked at the tumor size. So what I hope you can appreciate here is this is normalized to the control treated with PBS. When treated with PBS, however, when you treat with our agent, you can see a 30% reduction in tumor size. Now, 30% might not seem like a lot, but I want to remind you that we are looking at a disease where the therapeutic landscape is really meager. Achieving 30% tumor reduction is, by any measure, significant in this particular indication. Additional supports came from the observation that the animals that were treated with our biologic benefited from survival, basically. They had an extended life. So you can see that animals that received therapy live much longer compared to the animals that did not receive therapy. So this was very encouraging. Then we wonder whether this response in tumor size and longevity was due in part to the immune system. So I've showed you that the biologic can generate direct cell killing, but we also thought that maybe the biologic is also treating this immune component, so you get sort of this synergy between the two mechanisms. What I am showing you in C, so we interrogated basically the spleens of the animal that were treated to see if we can see some activity, some immune activity, and ensure we can see the activation of CD8 T cells in the spleen. And then when we went to the pancreas, we saw similar, at least for helper cell, CD4 cells were really also activated. We did see an increase of CD8, but that was really transient. And shortly after that, we can see actually a depletion of the activated CD8 cells into a microenvironment. So what we decided to do then was to further modify this biologic, this bacteria, to then give it the ability to release small molecules that can inhibit checkpoints, right? So to block again the PD-1, PD-L1 signaling cascade. So we produce a small RNAs that target a very specific molecule that control that PD-L1, PD-1 signaling. And you can see we can achieve that as a control. When we treat with iStorm, and this is the new agents, you can see we can reduce PD-1 expression. All right, so then to really test this, we decided to turn to a predictive model, right? So this was an orthotopic model, right? So we decided to turn to a genetic and predictive model of pancreatic cancer, which is the KPC. We first established the routes and dosage and toxicity, so I'm not gonna bore you with that, but it's there if you wanna see it. And using that, then we then established that a 510 to the 7 IP or IV was the best route. So treating this animal here, first we assess whether we can achieve tumor targeting in this translational model. Again, you can see after the first dose, we get really nice localization enrichment in the pancreas, which are shown at the very bottom. And even after the last dose, so remember we give 4 doses every 3 days, right? So it's 1 dose every 3 days, so total 4 doses. We can see that even after full dose, we do get localization again into the pancreas, enrichment in the pancreas. We do see this enrichment in the liver, but it's not surprising because the liver is a clearing organ. okay? And we did send these livers to pathology where they noted some inflammation and so forth, but not any significant drastic or concerning disease. And then our evolved strain shows similar behavior, either after the first dose or after the full dose. So we can, even after repeated challenge, we can still target the tumor. And that's very encouraging. Now in this model, we then ask, are we generating immune activity with our agent? So what I hope you can appreciate here, in A, you can see that the parent strain can activate some the CD8 T cells, and then if we separate by male and females, there seems to be a bias, sex bias, but we don't really think it's there. We can talk about that another time. And then we compare our agent, which is the new one here, which you can see in blue, which was evolved to generate checkpoint blockade molecules. You can see a dramatic increase in the percentage of activated CD8 T cells. I should say, if you can tell from the tone of my voice, I was very excited. Now that we show that we have a strain that can generate this really amazing response in the tumor microenvironment, we want to now use that strain as the lead agent that we can now further evolve to assign additional characteristics. And we can see also in the tumor response here, so again, here is your control. When we treat with PD-1, which is again current immunotherapy, these animals don't even respond. In fact, the tumors get even bigger. And when we treat with the parents or our strain, the new strain, we can achieve tumor suppression. So what we're currently doing, we are now combining this agent with checkpoint blockades, right? With now immunotherapy just to augment even further. And the standard of care, we're using chemos and so forth. And I should also say that using some omic-based approach, we identify potential candidates that we can use in combination with this therapy to achieve deeper response and durable response. Moreover, we're now doing sort of this longitudinal immuno-omic experiment to understand just the biology. What is the strain doing to the immune landscape in the tumors? Right? So we can't really move forward without that understanding. And then second, we're hoping that whatever information we can derive from there can then be leveraged to identify vulnerabilities and target those vulnerabilities to generate even deeper response. All right, so in summary, we know the pancreatic cancer space, again, is really terse in terms of therapeutic options. So we think that we have a unique and safe technology that can address the unmet clinical needs. And we hope with what I've showed you that we can generate sort of tumor localized immune activation. And this is particularly important in the setting of metastatic disease, where you could treat one lesion and that treatment will generate, so to speak, an abscopal effect where all these immune cells that are activated at that particular lesion can have more systemic effect throughout the patient. And I should also note that this technology does not rely on a priori knowledge on some very specific mutation or alleles that you have in the tumors. Current therapies, we need to first interrogate the molecular landscape of that tumor, identify actionable mutations, and then use a very specific agent against that specific mutation. And I've showed you that not all tumors are the same. You have so much genetic diversity, and we don't have agents against all these different oncogenic alleles, right? So we need to think, for example, in the case of pancreatic cancer, we need to think of ways we can target cancers to generate benefits more broadly, not just for a subtype of a molecular subtype of disease, but more broadly. And we think that with this technology, because we're recognizing features that are common across multiple types of cancers, we might be able to achieve that. And I will also note that the platform, right? So we're using a genetically programmable platform, and you have so much you can do there. And you can even load it with, you can load up to 300 KB of exogenous DNA into this strain.
So just imagine the power that you have.
With this technology. It is just remarkable. And I should also note that we think that it's also readily scalable. So we're currently talking to some colleagues where we can actually generate a lyophilized version of this technology so we can achieve scalability. So if it's lyophilized and reconstituted afterwards at the site, so that's easy, we can easily scale this for production. And this has an implication in terms of cost. Current immunotherapy treatment now, we're talking about $50,000 to $100,000, right? So with this part here, we think we will be able to then generate sort of a product that's a little more cost effective. And I should note that this is a technology that's under patent consideration. Okay, so and I will want to part with these thoughts, sort of this idea in the sky, this big pie in the sky, if you like pie or ice cream. So we know that we can utilize genomic editing to generate new behavior into this therapeutic strain, right? We've done that already. But what we think is even more powerful is actually integrating system biology approaches into these agents to really now integrate these really complex behaviors where we can program when the bacteria reach a certain sort of density and they self-destruct, right? Or we can program them to express a very specific molecule at very specific schedule. Like express this, and once you're done expressing this, express this. When you're done expressing this, then express that, right? So that is really powerful in terms of having to apply that type of system biology approach onto an agent that can clearly go to the tumor, right? So we are working with Paul, who's here, and Jorge as well, together to be able to enable that, to bring that to fruition here on this campus. And I will end by thanking all the wonderful people that made these studies possible. I want to highlight a search of people that were directly involved in this work. Melika, a graduate student in the lab, worked long hours in the animal room, so much that you can actually blindfold her and she can tell you where to go next. Durai was been in lab and soon will be going to a better life, I should say. And a few undergraduates, including Jacob Moyer, Nadia Patterson, who was a graduate student that just graduated from our lab, Rob, Katie, and Mwanza, which is a Fulbright faculty that came to work in our lab from Johannesburg, from South Africa. And I also want to thank my collaborator, especially Mario, who has been really nice in sort of in talking about his experience with his clinical trials with VNP, and my co-authors and other collaborators, including Misha Beltran, who have had extensive conversations on our pancreatic cancer, prostate cancer sort of indication. And I also want to thank Brian in the tech transfer office. He's been really instrumental in really laying out some of the things that we need to do to accelerate, at least moving this to market in some reasonable way, and make sure that we cross all our T's and dot all our I's. And I'll end by thanking you. But before I thank you, I should also thank the people that give us money. So all the people, all the icon. So I want to thank Embark here, Midwest Biomedical Accelerator Consortium, which is a partnership with the NIH REACH program on moving technology from the bench to the bedside, the NIH, the American Association for Cancer Research, and the University of Missouri for support. Thank you.
Thank you so much. So we'll start with some questions, and one online that came in was curious why you chose salmonella.
I don't know that it was really a choice, but so maybe I should say this. So when I came to Colombia, when I started my position here, I was approached by Dr. Abe Azenstar, who was passed on now, and I know he was working with this technology. And I immediately saw the promise. I saw the potential, again, just to leverage its tumor targeting capacity to achieve broad targeting. So it's not so much that I had the library in front of me that said, oh, I have salmonella, I have E. coli, I have lactobacillus, and so forth. And I said, I'm gonna go with salmonella. So it's more sort of things happened. Okay. Yeah, that's honest. I couldn't make it fancy story by that.
Yeah, it was great presentation. I really enjoyed. I have two questions, very quick ones. So one of them is that you mentioned that your bacteria, or your delivery vehicle, it caused the death in the tumor cells. So it caused. So what type of cell death do you observe? Is this necrosis? Is this?
That's beautiful, beautiful question. So the question, that if no one heard it, so how is the agent actually killing the cells that it colonizes? So we think it's actually causing mitochondrial catastrophe. So we can actually see progressive mitochondrial degeneration from the cisternae. So we have EM data that shows a following invasion, and that causes ROS and that causes cell death. So we think it's ROS dependent cell death. That's a beautiful question.
Okay, another question is very naive. So since you're putting all these bacteria into the animal, or hopefully soon into the patients, so how do you think that this will have impact on the normal microbiome?
This is great. So the irony behind that question is I asked the same question last time when Paul was presenting. So that question is turned back to me. So we think, no, it's okay. no. So that's it. It's just ironic. So we think it's certainly going to have an impact, especially if you administrate it IV, right? You're certainly going to have an impact. I think we don't know what that impact will be, but we know that bacteria and the microbiome in general can influence immune response, right? So that will have to be empirically established.
Hi. Great talk. Lots of exciting stuff coming there, Yves. Thank you.
Thank you.
My question is about pre-existing immunity, friend or foe, or both. Mm-hmm. So I could imagine that there are people, or maybe dogs, that have antibodies already to salmonella before the experiment or the disease begins. And would you see that as possibly inhibiting the therapy, or maybe helping it if there's a pre-existing memory response and it makes it to the tumor? Is that an advantage? And related to it, which is really the same question, the human trials that you said had problems, do you happen to know if they pre-screened to select people for that trial so they don't have pre-existing antibodies or something like that? Anything you think on this issue? Thank you.
Great questions. So I'll start with the last question. I'll address the last question first. So in the enrollment criteria, the patient could not have pre-existing immune-based conditions. So that was definitely an enrollment criterion. Second, the question of pre-existing immunity. So we think that is definitely going to bias, that is going to affect the treatment. So if you treat and you have memory cells already there and then your agent basically get mopped away, that will affect how you dose and that will affect the efficacy, right? So I think as a proof of concept at the beginning, I think we can learn also from Mario's clinical trials and Bermud's clinical trials. And when you're selecting patients, you have to select patients that do not have pre-existing immunity against salmonella. But that's a good question.
And this will be our last question.
You are using the metabolic complementarity to enhance the tropism of the bacterial agent to the cancer. I wonder if you can extend that on enhancing the anti-tumor action by by malnourishing the tumor. I think by hopefully depriving the tumor of micronutrients essential like iron, zinc, or essential amino acids. And I might have a second question that would be related with the resurrection of the immune system, if that will imply the clearance of the bacterial agent that have started the immune killer. I mean,
Great. So the first question was, can you leverage the tumor tropism to basically deliver some metabolite or something that's toxic to the tumors? So conceptually, yes, that's feasible, but we're not necessarily looking at that for now. And then the second question was,
the resurection of the immune system
sorry.
against
Yes,
the tumor
yes.
would bring
So-
the clearance of the
Yes.
bacteria itself, and
Yes.
also the tumor,
So-
and kind of diminish.
Certainly, certainly. So the question is whether treatment-induced immune resurrection, to use your own words, that can actually lead to the elimination of the therapeutic agents. That's what you're saying. Yeah, I think that'll have to be empirically, right, tested. See, in the first challenge, second challenge, do you need to increase the dose as you move on, right? But based on what we saw earlier, right, after multiple challenges, you're still able to get some targeting. You might not get the same targeting that you had at the first injection, but if you get some targeting and some therapeutic activity that can trigger immune activation, that's what you want, right? So,
Thank you again. Thank you so much. Let's give him another round of applause. We hope to see you all next month for Dr. Koopman in December.